Hey everyone! So let's get back to microbiology and let's go ahead and start with chapter 3. You may have noticed that we skipped chapter 2. Chapter 2 is some basic chemistry, and I tend to teach chapter 2 as needed as I go through the other chapters. So chapter 3 is all about prokaryotic cells. We will be talking about the anatomy, what makes up a prokaryote, the different features that a prokaryotic cell will have. We're going to talk about the cell wall. We're going to talk about the plasma membrane. We're going to talk more about Gram stains and acid-fast stains and how they work based on the structure of the bacteria. We will close the chapter by talking about endospores. We mentioned them briefly in the last chapter because we were talking about how to stain them. Well, now we're going to talk a little bit more about the sporulation process and just give you some more information on endospores in general. All right, so before we begin, let's just start talking spend a few minutes talking about prokaryotic cells versus eukaryotic cells. So they're very, very different cell types. They do not share that many similarities. So if you remember, prokaryotes are the bacteria and the archaea, whereas the eukaryotes are pretty much everything else. So they really only have 3 common features between the two cell types. Yes, both cell types have DNA, and that DNA is found in chromosomes. However, it's packaged differently. In prokaryotes, the DNA is found in a circular chromosome. So that's right, prokaryotes typically have one circular chromosome. Now there are some species of prokaryotes that have 2 chromosomes or 3 chromosomes, but for the most part, most bacteria, most prokaryotes have 1 chromosome and it's circular. If you compare that to eukaryotic chromosomes, well, eukaryotes such as ourselves, our chromosomes are linear. And we have many more than just one. Okay, we have 46 chromosomes, we have 23 pair, but that differs among the different species of eukaryotes. So both cell types also have a cell membrane. You probably learned this in anatomy and physiology. It was called the phospholipid bilayer. So the cell membrane, we'll call it both cell membrane. Sometimes I may make a mistake and call it a plasma membrane, but it's really correct terminology as well. But this is that phospholipid bilayer, and we'll talk more about that. Both cell types have cytosol, that is the liquid inside the center of the cell, and both cell types have ribosomes. So ribosomes, if you remember, this is the site of protein synthesis. That's really the only similarities between prokaryotic cells and eukaryotic cells. Okay, so now let's talk about some of the distinctive features. Well, with prokaryotes, and if you see in the upper right hand corner, that is a typical bacteria cell. Okay, so prokaryotes, remember, they're extremely small. They do not have a nucleus. They do not have traditional organelles, with the exception of ribosomes. I mentioned that they have one circular chromosome. It is not membrane bound. Therefore, there is no nucleus. Very important. If you look at the purple structure in the center of that bacteria cell, that is its chromosome. Now, it says it's a circular chromosome. If you were to take that purple material out and actually spread it out, it would be circular. In this particular bacteria cell, you can see where they kind of crammed it all inside. That is the normal structure of the chromosome at any given time point. Like I mentioned, no organelles except for ribosomes. They do have peptidoglycan cell walls. And remember, they do not undergo mitosis or meiosis. They reproduce through a process known as binary fission, and we'll talk more about that in just a second. So let's talk briefly about binary fission. I mentioned in the last chapter, It's a very simple process. It's simply where one cell divides into two cells. It is considered asexual reproduction because there is no exchange of genetic material. So the new cells, or the offspring, should be genetically identical to the parent cell unless some type of weird mutation took place during the copying process of the DNA. So if you look at the steps of binary fission, the DNA is copied, the cells grow, the copied chromosome is pulled down to one end of the cell, and then a septum forms. The septum is a partition that separates the one cell into two separate cells. Okay? And then it pinches off, so you wind up with two cells. So if we look at the next slide, you can see that in picture form, I really like pictures, you see it starts off with one cell, one chromosome. The chromosome replicates, the chromosome is pulled down to one end as the cell starts to elongate, the septum forms, and it winds up pinching off into two separate cells. Binary fission is considered to be exponential growth because one cell yields two cells yield four cells yield 8 cells, and so on and so forth. Binary fission can be an extremely fast process. For example, E. coli cells reproduce roughly every 20 minutes. Okay, so definitely a much faster process than any type of mitosis or meiosis. That's one of the reasons that some bacterial infections can get so severe so quickly, is because they reproduce very quickly. All right, so moving on to size, shape, and arrangement of bacteria. The average size of a bacteria cell, yes, it can range quite a bit, but it says roughly 2 to 8 micrometers. Okay. On average, I would say 5 micrometers. So again, there is variation because some bacteria, as we will learn in just a minute, are longer than they are wide. Okay. Whereas other bacteria are perfect spheres or perfect circles. And so again, there is a little bit of a variation between size, but on average, I would say 5 micrometers is a true average of bacteria. Although the mycoplasmas are a very unique class of bacteria. They are incredibly small. They are some of the smallest known bacteria. And in fact, for a long time, we didn't know that these even existed because they're so small they cannot be seen with a light microscope. We didn't discover these until the invention of the electron microscope so that we could actually see these really, really small bacteria. Bacteria, for the most part, come in 3 basic shapes. Bacillus cells, okay. If you look at the picture to the right, that is the shape of a bacillus cell, okay. They're also sometimes referred to as rods, or typically I just call them sometimes hot dogs. They're hot dog shaped cells, okay. Moving on to the coccus cells, or cocci if it's plural. These are bacteria that are round. They are round spheres, okay. And then the third group of bacteria are your spirals. And within the spirals, this group can actually be broken down into 3 subgroups: the vibrios, the spirillums, and the spirochetes. And we will go over those 3 different classifications on the next slide. Okay. Most bacteria are monomorphic. Some bacteria are pleomorphic. Okay. So if we look at the word monomorphic, the prefix mono means one, morph means shape, so one shape. Most species of bacteria are monomorphic, meaning they always have one shape. They always look the same. Again, if you look at that picture on this slide, those are monomorphic bacteria. They all look the same. In a few minutes, I do have a slide where I will introduce pleomorphic bacteria. With pleomorphic bacteria, they do not look the same, okay? And they are more on the rare side as far as species are concerned. There's not a whole lot of species of bacteria that are considered to be pleomorphic. We will also talk about cell arrangements, some of the different cell arrangements that you can find for the coccus cells and some of the different cellular arrangements that we will find for the bacillus cells, or those rods. So let's go and talk more about the spirals. So I said the spiral bacteria could be broken down into 3 categories. You have your vibrios, your spirillums, and your spirochetes. Your vibrios, they kind of look like a boomerang. They're short, they have fairly thick cell wall, they have a slight curve to them. They kind of remind me of a Cheeto Puff or something like that. Your spirillums are longer than your vibrios, but they're shorter than your spirochetes. And they typically have one complete 360 curve in there, or an alpha helix structure in there. And then lastly are your spirochetes. We talked about the spirochetes in the last chapter because I talked about how they did not stain very well due to the nature of them having, they're really long, they're thin, and they have a corkscrew appearance, and so therefore they don't take up stains very well. They have that helical structure. Now the spirochetes are very interesting. They are able to move. They're able to swim forward, they're able to swim in reverse, they're able to curl up, and then they're able to expand back out. I say that the spirochetes remind me of earthworms. If you've ever watched an earthworm move, they have a variety of different movements that they can make. The spirochetes are very much like that. And again, there will be a video link that I want you to watch that will show you spirochete movement, just so you can see how well that they move. Okay. All right, so when I was talking about monomorphic bacteria versus pleomorphic bacteria, well, I said most species of bacteria are monomorphs, okay? One shape. One cell looks identical to the neighboring cell, such as the example here with E. coli. All of those cells, they look the same. They're all bacillus cells, or those rod cells. Whereas if you look at the pleomorphic bacteria, many shapes, those cells do not look like each other. Some cells look like vibrios. Some cells look like rods. Some cells are thicker at one end than at the other end. So there's a lot of variation with the pleomorphic bacteria. And again, the pleomorphic bacteria are more on the rare side. Moving on to cell arrangement. So this is different arrangements that these bacteria cells can be found in. If we look at the left side of the slide first, we look at the coccus cells. So if you have a single coccus, it's literally just called that, a single coccus cell. Some bacteria species present themselves as single coccus cells. Okay. However, sometimes some species will present themselves as being diplococci. Again, di meaning two, so it's where you have two cocci that are stuck together. Next, A is your streptococci. Well, we learned back in chapter 1 that the prefix strep means chain. So your streptococci is literally a chain of coccus cells. If you look at B, you see a tetrad. The prefix tetra means 4. So a tetrad is where you have 4 bacteria cells, and they are stuck together. But notice, they are organized. It's not like they're just randomly smushed together. They are organized in a nice layer. Next, if you move on to the Sarcinae. The Sarcinae, well, it looks like you have 2 tetrads stacked on top of each other. Again, it's a nice orderly fashion. And then the last grouping are your Staphylococci. Well, if you remember from the last chapter, I mentioned that the prefix staph meant cluster of grapes. So your Staphylococci, literally, it's a cluster of cocci. And there's no nice, neat arrangement with the Staphylococci. They're just all smushed together to represent that cluster of grapes. Now, if you look over to the right, we see different types of cell arrangements with the bacillus cells. You see in A, single bacillus. Okay. Some bacteria species present themselves as diplobacilli. So again, 2 bacillus cells stuck together. We also have streptobacilli, just like we had streptococci. Strep, anytime you see strep, it means chain. So streptobacilli means chain of bacillus cells. Okay. And then lastly, this is kind of a weird one. These are known as the coccobacillus cells. They have characteristics that are kind of like coccus cells, but they also have characteristics that are similar to the bacillus cells. They're more oval shaped than anything else. So these are a very unusual, weird group. These are kind of like the outliers. They don't fit nice and neat into the coccus arrangements or into the bacillus arrangements. One thing I want to point out is planes of division. If you notice, there are more cell arrangements with the coccus cells than with the bacillus cells. And if I were to ask why, well, the reason being is it comes down to the number of planes of division. Coccus cells can divide on the side or on the top, so they have 2 planes of division, whereas the bacillus cells only divide on the side, so they have 1 plane of division. So again, coccus cells have 2 planes of division, bacillus cells only have 1 plane of division. And so because of that, there's more types or variety of cell arrangement when it comes to the coccus cells than when dealing with or when referring to the bacillus cells. Okay, so now let's move on and talk about the structures that you can find on a bacteria cell. So we're basically looking at bacteria anatomy here. We are going to start on the outside of the bacteria cell, and we are going to work our way in to the inside, to the innermost area of the bacteria cell. All right, so here's a list of things that could potentially be found on the outside of a bacteria cell. Notice I said it could potentially. There are going to be some species of bacteria that do not have any of these structures. There will be some species of bacteria that may only have one of these structures. There may be species of bacteria that only have 2 or 3 of these structures. So it really depends, and it varies quite a bit on the species of bacteria that you're looking at. So we are going to talk more about each of these, the glycocalyx, the flagella, the axial filaments, the fimbriae, and the pilus or pili. So let's start by talking about the glycocalyx. So the glycocalyx is made on the inside of the bacteria cell, and it's excreted through the cell wall to the outside of the bacteria cell. Now, like I just said, remember, some bacteria have a glycocalyx, some bacteria don't. Again, it depends on the species. The glycocalyx is sticky, and if you look at the word glycocalyx, the prefix gly means sugar, and sugar is typically sticky. So that's one way of helping you remember that it's sticky, and the fact that it's sticky will help you remember what it does. So the glycocalyx comes in 2 forms. Sometimes the glycocalyx will be in the form of a capsule. Other times the glycocalyx will be in the form of a slime layer. So it doesn't matter, capsule or slime layer. Either way, it's composed of the glycocalyx. It's made of glycocalyx. So what is the difference between a capsule and a slime layer? Well, in a capsule, the glycocalyx is organized, it's firmly attached to the cell wall, it's uniform, it's not patchy. You can see a picture of the capsule on the right, and you can compare the picture of the capsule to that of the slime layer. Because when it comes to the slime layer, it's still glycocalyx, it's still sticky, but with the slime layer, it's unorganized, it's loosely attached. There may be patches on the outside of the bacteria cell where the slime layer is missing, so it's patchy. So that is basically the difference between the capsule and the slime layer. It comes down to the organization of the glycocalyx on the outside of the bacteria. We talked about capsules in the last chapter because we talked about how they don't stain very well. They're this gelatinous material. They're sticky. They don't stain very well. Well, you can see a picture, the upper right-hand corner. That is a picture of bacteria cells that have a capsule around them. So what does the glycocalyx do? Well, the glycocalyx has two functions. It allows for cells to attach, which is key to biofilms and biofilm formation. It also helps prevent phagocytosis, which is a virulence factor, meaning it allows the bacteria to surpass our immune system. So let's talk more about those. Allows cells to attach, key to biofilm. Well, first of all, what is a biofilm? A biofilm, an example of a biofilm is pond scum. Okay. If you've ever seen a pond and there's this layer laying on top, that we typically call it pond scum, that is a biofilm. A biofilm typically starts off as one single cell that has a glycocalyx. And because the glycocalyx is sticky, it's able to bind to neighboring bacteria cells. And not only does it bind to neighboring bacteria cells, it also can bind to rocks or grass or vegetation, glass. It is able to bind to a variety of different substances. And that is why the pond scum layer can get so big. So not only can it get large in size, but a biofilm can get 300 to 500 layers thick. Okay, so we'll be talking more about biofilms later this semester because it turns out they play a big role in hospital-acquired infections. But other examples of biofilms, just to kind of give you an idea, if you ever scrape your teeth and you get plaque off of your tooth, that is a biofilm. If you have a shower curtain, if you feel your shower curtain and you feel it's got like this soapy scum, we call it soap scum, that's a biofilm. If you wear contact lenses, biofilms form on the surface of contact lenses, which is why you're supposed to make sure you clean contact lenses on a regular basis. So that is basically what a biofilm is. It starts with a single bacteria that has a glycocalyx, and it's able to bind to neighboring bacteria that also have a glycocalyx. And so the one cell binds to neighboring cells, and before long, you've got a billion cells that are now in this biofilm because there's the glycocalyx present in so many of these cells, and it's sticky and they're able to bind to one another. All right, so okay, moving on to prevents phagocytosis. All right, so to recap phagocytosis, I think you learned about this in A&P, but just in case you didn't or you need a recap, part of your immune system is composed of cells called phagocytes. Phagocytes are known as cell eaters. They circulate throughout your body, and when they come upon a cell or something foreign that's not supposed to be there, they wrap around the foreign entity, they engulf it, and then they break it down. So if the glycocalyx is present, those cells cannot be phagocytized. So say you have a pathogenic bacteria that enters your body, your immune system says, oh no, that's not supposed to be here. Let's engulf it and break it down. But if that pathogenic bacteria has a glycocalyx around it, then those phagocytes cannot engulf it and break it down. So now your immune system has to send the next level or the next layer of your immune system to try to attack these cells. So the key point, these cells, if they have the glycocalyx around them, they cannot be phagocytized. So it is a way of the bacteria surviving your immune system. And that is what is meant by virulence factor. There are quite a few bacteria that have, a pathogenic bacteria that have capsules. Because if you think about it, the bacteria, the goal of the bacteria is to reproduce. Well, a lot of these bacteria, if they're pathogenic, they have to be able to get into your body and stay there, stay in a particular location. The glycocalyx helps them do that. Because of the glycocalyx sticky nature, you inhale pathogenic bacteria, and the stickiness allows the bacteria to get stuck in your nose or the back of your throat or your respiratory system. So more often than not, pathogenic bacteria tend to have a glycocalyx. Okay. So at the bottom, you see some examples: Bacillus anthracis, again, the bacteria that causes anthrax; Streptococcus pneumoniae, which causes a type of pneumonia; S. mutans. So I mentioned plaque a minute ago. S. mutans is a bacteria that is commonly found in your mouth. It is the #1 cause of tooth decay or of cavities. And again, it has a glycocalyx around it. So that sugary substance helps that bacteria to bind to your tooth, promoting tooth decay. So that's a little bit about the glycocalyx, what it is, what it's composed of, and what it does. All right, moving on. Okay, so moving on. Now let's talk about flagella. Roughly half of bacteria cells have at least one flagella. You may remember that flagella are used for locomotion. They help the bacteria move or swim through the environment that they're in. A lot of students tend to think that bacteria just sit still all the time. That is not the case. Most bacteria move. They are swimmers. They are able to move very, very well. Okay? And in order to do that, they need some type of appendage to help them move. And so the flagella is one of the largest categories of appendages that help these bacteria move through water, through a watery environment, through your body. All right, so if the bacteria cell has a flagella, the flagella is firmly attached to the cell wall. That becomes important on the next slide. Just keep in mind that the flagella are firmly attached to the cell wall. Number and placement, well, there can be a variety of different placement types and numbers. So the first term is atrichous. The prefix a means without. Trichous literally translates into hair. So atrichous means without hair. But we are referring to flagella here. So atrichous means without flagella. So if you have a bacteria cell that does not have any flagella, it is called atrichous. The next one, monotrichous. Well, mono means one, so monotrichous means one hair or one flagella. The picture in the top right corner is a bacillus cell with one flagella, so it is monotrichous. The next term, lophotrichous. Lopho means tuft, T-U-F-T, tuft, like a tuft of hair. So the one in the middle, the picture in the middle on the right-hand side, you see a bacillus cell, or it's kind of more of a vibrio actually, and you see it has a tuft of flagella at one end. So that is lophotrichous. Amphitrichous. The prefix amphi means both. So if you look at the picture on the very bottom on the left, you have a spirochete, and you can see that it has a flagella on both ends. That is amphitrichous. And then the last term is peritrichous. The prefix peri means around or surrounding. So if you look at the picture on the bottom right, you see a bacteria cell and it has all kinds of flagella coming from all around it. So again, that is an example of peritrichous. Okay. So let's talk about flagella motility. Well, first of all, flagella are composed of a protein known as flagellin. Okay. The bacteria move due to the rotation of the flagella. Now keep in mind, I said on the previous slide that the flagella are firmly attached to the bacteria cell. So the flagella rotate clockwise, and it allows the bacteria to move along or swim along. Well, that flagella, because it's firmly attached to the cell wall, it can only rotate so much before it simply gets stuck and it stops and it can't rotate anymore. Think of winding a clock. You can only wind that clock so far before you can't wind it anymore. It's the same way with the flagella. So as this mechanism is known as run and tumble. So the mechanism of rotation of how bacteria move is known as run and tumble. As the flagella is turning clockwise, the bacteria are running, or they are moving. And then at some point, that flagella rotates to the point it can't rotate anymore. So it stops, it gets stuck. And at that point, that is when the bacteria comes to a dead stop. And it almost looks like, if you watch bacteria move, it almost looks like they tumble. Okay. And during the tumble process, what's happening is the flagella is reorienting itself back to its original position. And then it will get ready to run again. So the bacteria cell will alternate run and tumble, run and tumble, run and tumble. Bacteria do have the ability to move toward a stimulus or away from a stimulus. This is known as taxis. Just a couple of examples. Chemotaxis is when there is food present and the bacteria is going towards the food source. That is chemotaxis. Photosynthetic bacteria need sunlight. So if bacteria are moving towards the sunlight, that is phototaxis. Aerotaxis, if bacteria are moving towards sources of oxygen, okay, that is aerotaxis. And magnetotaxis. We haven't talked about this yet, but we will. Certain types of bacteria require iron in very large amounts, okay? So if the bacteria senses that there are iron stores present, they will go towards that iron. That is known as magnetotaxis. Here you can see the run and tumble mechanism. If you look at picture A, you can see the run and tumble. There's no attractant or repellent. There's no type of taxis going on here. So again, they're just kind of doing their own thing, run and tumble. Well, if you look at B, there's a gradient. There's something present on the far right. Maybe it's a food source, or maybe it's sunlight. And the bacteria are swimming. They're trying to get over to that attractant. So they still go through the run and tumble process, but you can see how they actually have direction now. They're actually moving to that stimulant or to that attractant. Okay, so that is run and tumble. All right, so we are done talking about flagella. So let's move on and talk about another potential external structure, and this is the axial filaments. Well, let me go ahead and tell you to go ahead and highlight this, make a little note. The second bullet under axial filaments. Axial filaments are only found in spirochetes. They're not found in coccus cells or bacillus cells or any other type of cell. They are only found in spirochetes. And the reason for this, they're known as endoflagella because the axial filament is firmly attached to one end of the spirochete, and then it wraps around the entire length of the spirochete and it firmly attaches on the other end. It is due to the rotation and the movement of the axial filament that causes this spirochete to move. Remember, I said that they have earthworm-like abilities. They're able to swim forward and swim in reverse, and they're able to stop and curl up and expand back out. That's all due to the rotation and the movement of these axial filaments. So the axial filament, again, only found on spirochetes. Now, if you move over to fimbriae, so fimbriae also allow for attachment. They are short, bristle-like protein structures on the cell surface. They work alongside the glycocalyx in forming biofilms. So again, very important in biofilm formation. You can see they don't really look like flagella because they vary in length. Some are really, really short, some are much longer, but those are fimbriae and they tend to be very common in gram-negative bacteria. All right, moving on to the last structure that you could potentially find on the outside of a bacteria cell are the pili. Now, typically if a bacteria cell has a pilus, they only have one. Some bacteria cells have been seen to have two pili. But for the most part, most bacteria, if one is present, they only have one. So pili, somewhat similar to fimbriae, less numerous and more rigid. Pili are used for the transfer of genetic material from one bacteria cell to the next. Now remember, when it comes to bacteria division, I said they divide through binary fission. There's no mitosis or meiosis. There's no exchange of genetic material. But with the pili, this is one mechanism that does allow for the exchange of genetic material. This process is known as conjugation, or the common name would be bacterial sex. So here you see two cells, and the cell on the left is the donor cell. It has the pilus or the pili, and it literally injects its pili into the recipient cell, and it's able to spread genes through the pili from one cell or from the donor cell to the recipient cell. These some of these genes code for things like antibiotic resistance. So if the bacteria cell on the left is resistant to tetracycline, if it inserts its pili into the neighboring recipient cell, it can potentially pass that gene that codes for tetracycline resistant onto that recipient cell, making that recipient cell also resistant to tetracycline. Okay. So antibiotic resistance is spreading via pili. And so again, pili is used for the transfer of genetic information. All right, moving on to the cell wall. Okay, so we are done talking about external cell structures. We have now moved on to the cell wall. Remember, pretty much almost all bacteria have a cell wall. Now there is a group of an exception, and we will talk about the exception in a few minutes. But again, I would say 95% of bacteria do have a cell wall. The purpose of the cell wall is to give the bacteria shape, structure, and to help protect it, help protect that bacteria cell from busting open and breaking down. So the cell wall is very, very important. We learned in chapter 1 that the cell wall is composed of something known as peptidoglycan. If we take that structure of peptidoglycan, we can actually break peptidoglycan down into its substructures. So if you look at bullet 2, the cell wall, it says the cell wall consists of peptidoglycan, and in parentheses it says murine. Older textbooks and some newer textbooks will use the term murine instead of peptidoglycan. But it consists of two polymers that are carbohydrate in nature. One is known as N-acetylglucosamine and the other is known as N-acetylmuramic acid. These are commonly just abbreviated NAG and NAM. Okay. And so on the next slide, I have a picture of the cell wall, and you will actually be able to see how the NAG and NAM come together to form the backbone of the peptidoglycan. So the NAG and NAM, they alternate. So you'll have a NAG unit and then you'll have a NAM unit right next to the NAG. So you'll have NAG followed by NAM, NAG, NAM, NAG, NAM, and so on and so forth. You'll have these really long strands of carbohydrate backbone of NAG and NAM, alternating subunits of NAG and NAM. In addition to the NAG and NAM, you have polypeptide cross bridges and you have tetrapeptide side chains. All right, if you remember, what is a polypeptide? Well, a peptide is an amino acid. So a polypeptide, this just means a small chain of amino acids. Okay? And they form cross bridges. So the polypeptide reaches across those carbohydrate backbones to help hold those carbohydrate backbones together. And then you also have tetrapeptide side chains that are attached to NAM. Well, if you look at that word tetrapeptide, tetra means 4. Peptide is an amino acid. So it's 4 amino acids. And they form side chains that are attached to NAM. So what really makes up peptidoglycan are these alternating subunits of NAG and NAM, followed by cross bridges and tetrapeptide side chains holding those carbohydrate strands of NAG and NAM together. Now, before we move on, when you hear the term cell wall, it's very easy to picture a wall in your house or a wall in the building. A wall is typically a very solid structure that does not allow any type of material to pass from one side of the wall to the other side of the wall. Well, that is not the case with the cell wall. The cell wall, this peptidoglycan that we've been talking about, is very loosely woven so that materials such as ions, amino acids, and small sugars are able to pass through the cell wall from one side of the cell wall to the other side of the cell wall. Okay. Okay, so here's that picture that I was talking about. Again, I'm a picture person. I really like pictures. So this is peptidoglycan, the cell wall, broken down into the subunits. So I said that the first thing are the alternating subunits of NAG and NAM form these long carbohydrate backbone strands. So you can see that it's that light pinkish purple color as well as that darker pinkish purple color, and they're labeled NAG and NAM, and you can see those strands will continue NAG, NAM, NAG, NAM, NAG, NAM, and so on and so forth. Coming off of NAM, you see the tetrapeptide side chain. It only attaches to NAM, never NAG. Okay. And then you see the polypeptide cross bridge in blue. So the cross bridge does exactly what the name says. It crosses over to form a bridge, and it helps hold those long strands of NAG and NAM together. Now, if you look, also the term peptide bond is labeled. A peptide bond that's simply the name of the bond that holds 2 amino acids together. So all of those spheres that are in green and all of those spheres that are in blue, those are all amino acids. So holding any 2 amino acids together is a peptide bond. Okay. All right, so this is peptidoglycan in its most basic form, and you can see again how it's loosely woven so that ions and small sugars can easily pass from one side to the other side. Okay, now let's compare and contrast gram-positive cell walls versus gram-negative cell walls. And we touched on this in the last chapter because we were talking about gram stains and how gram stains worked. Well, remember, gram-positive cells have a very thick layer of peptidoglycan. Okay. They do have an inner plasma membrane, or that inner phospholipid bilayer, which can be seen in yellow. But that very thick layer of peptidoglycan, you can see it's loosely woven, and you can still make out the strands of NAG and NAM and all of those peptides that are involved in forming the peptidoglycan. Now, typically on gram-positive cell walls, you find teichoic acids. Teichoic acids, all you need to know for now about teichoic acids is that it is an alcohol with a phosphate group attached to it. Now, if you compare that to a gram-negative cell wall, the gram-negative cell wall is very, very thin. If you look at this picture of the gram-negative cell wall, it looks like a single layer of peptidoglycan. Very, very thin. The gram-negative bacteria also have an outer plasma membrane, or an outer cell membrane. Okay? So that is basically the 2 main differences between gram-positive cells and gram-negative cells. Gram-positive cells have a thick layer of peptidoglycan, but they do not have an outer cell membrane. The gram-negative cell walls have a very thin layer of peptidoglycan, but they have an inner and outer cell membrane. So very thin layer of peptidoglycan, inner and outer cell membrane. The gram-negative bacteria are harder to kill. So when it comes time to treat an infection with antibiotics, some antibiotics will only kill gram-positive bacteria. Other antibiotics will only target gram-negative bacteria. Some antibiotics are known as broad spectrum, meaning that they'll kill both gram-positive and gram-negative. Okay, so keep that in mind, particularly with one of your homework assignments that you will have. It's a video that you will watch. It talks about how hard the gram-negative bacteria are when it comes time to kill them using some of the different antibiotics that we have. So now that we've talked about the cell wall, we've talked about the peptidoglycan, the different components that make up the cell wall, let's revisit the gram stain. And let's talk more in depth about the gram stain and how the gram stain actually works so that gram-positive cells are purple and gram-negative cells are that reddish pink color. So if you remember in the beginning, you use crystal violet to stain. It's the first stain that you use, and it stains all cells purple. Well, the next step is that you add your mordant, which is your Gram's iodine. What the mordant does is it binds to the crystal violet to form these crystal violet iodine crystals or complexes in the cell, and these are fairly large complexes. So now if you look at the gram-positive cells, remember that the gram-positive cells have the very thick layer of peptidoglycan. The third step of the gram stain mechanism is that you add your decolorizer, which is your alcohol. What the alcohol does when you add it to a gram-positive cell is it attempts to dehydrate the peptidoglycan, almost acting like shrink wrap. It dehydrates it, forming a tight seal, trapping the crystal violet inside the cell. So that is why the gram-positive cells with that thick layer of peptidoglycan are purple, because that purple dye gets trapped and held inside the cell due to the alcohol dehydrating the peptidoglycan. Now on the flip side, with the gram-negative bacteria, remember they have a very, very thin layer of peptidoglycan. So what typically happens is with the third step, the decolorizer, the alcohol, when you add the alcohol to a gram-negative bacteria, it can't form a tight seal. It can't trap that dye inside. What winds up happening is the alcohol winds up forming holes in that super thin layer of peptidoglycan. And now that there's holes, that crystal violet has the ability to leave. So at this point, that's why you have to counter stain your cells with safranin so that your gram-negative bacteria will show up. And it just so happens that with safranin, they show up that reddish pink color. So that is the gram stain mechanism and how it actually works on the cell based on the structure of the peptidoglycan. So I said that most cells have a cell wall. I think I said something like 95% of bacteria cells have a cell wall. Well, what about the bacteria that do not have a cell wall? Let me introduce you to the mycoplasmas. So these are the unique group of bacteria that lack a cell wall. Typically, they cannot be detected by regular light microscopy, because the cell wall is what stains. During the staining procedure, you're staining the cell wall. Well, if a bacteria cell has no cell wall, then there's nothing for the stain to bind to. So you cannot really stain the mycoplasmas. And if you can't stain them, well, then you can't use general or regular light microscopy in order to view them. If you look at this picture here, this is another example of pleomorphic bacteria. because they don't have a cell wall. And remember, one of the functions of the cell wall is to give the bacteria shape. So there's nothing giving the mycoplasmas shape. So one bacteria cell looks completely different than its neighboring cell because, again, there's no cell wall. So again, an example of pleomorphic bacteria with the mycoplasmas. Okay, we also talked about the acid-fast bacteria in Chapter 1. And we talked about how you cannot gram stain the acid-fast bacteria because the acid-fast bacteria had a waxy layer. And that waxy layer, I introduced you to the proper term, mycolic acid. So acid-fast bacteria have a layer of mycolic acid that covers the cell wall. The mycolic acid will not take up gram stain reagents. So that's why a different technique for staining the acid-fast cells was necessary. So again, this is just kind of a recap from the last chapter. There's two genera that are considered to be acid-fast, Mycobacteria and Nocardia. Both of these are of healthcare importance. For example, tuberculosis is caused by Mycobacterium tuberculosis. Leprosy is caused by one of the species of Mycobacterium. So those are both, tuberculosis is respiratory. Leprosy is actually spread through sneeze droplets as well. So it's actually considered to be respiratory as well. Nocardia is yet another respiratory disease that has about a 50% death rate. So both of these, Mycobacterium and Nocardia, these are very, very important in the health field. We needed a quick way of being able to stain them and differentiate them from other bacteria. So keep in mind that due to the mycolic acid, and you can see that layer of mycolic acid in the picture, it's represented by those purple squares. The mycolic acid is a pretty sturdy, solid substance that does not allow things like nutrients to pass very easily through that layer of mycolic acid. So because of that, the acid-fast bacteria tend to grow slower than non-acid-fast bacteria. Also note that different types of drugs, such as antibiotics, it cannot pass through that waxy mycolic acid layer. And so it's harder to treat these different diseases that are caused by acid-fast bacteria. All right, just a couple of ways. We will talk more in depth later on about damaging the cell wall, but just to kind of introduce you to a couple of ways of damaging the cell walls. Yes, you can damage the cell wall, that peptidoglycan, with lysozyme. So lysozyme is an enzyme. Okay, it's found in our tears. It's found in our saliva, because every time I open my mouth to speak, bacteria enter my mouth. Every time my eyes are open to the environment, I have bacteria entering my eyes. And what if they're pathogenic? So the function of lysozyme is to attack those bacteria, causing the destruction of the cell wall, which ultimately will cause the breakdown and death of the bacteria cell. Okay. So it's a protective mechanism. Also with penicillin. So penicillin is actually a class of drugs. There is one drug known as penicillin, but there's actually an entire class of drugs, and there's 50 different drugs in this class that are called penicillins, and it's based off of chemical structure, which we will get to later. But the penicillin, it works by preventing those peptide bridges from forming in the peptidoglycan. So basically, if the peptide bridges can't form, then the cell wall can't form, and therefore the bacteria cells will become very unstable and unsteady, and they will lyse or they will break open. Okay, next we are done talking about the cell wall. And remember, we started on the outside of the bacteria and we are working our way in. So the next thing that we come to is the cell membrane. Now, I've already made the mistake a couple of times of calling it a plasma membrane. It's also known as the phospholipid bilayer. Those three terms are interchangeable, but I do try to stick with what the book calls it, and the book tends to call it the cell membrane. So just a little bit about the cell membrane. It is called the phospholipid bilayer. Bi meaning two, because you have two layers of phospholipids, and we'll see this on the next slide. I have pictures. And the phospholipid bilayer is embedded with proteins, okay? And I'll point those proteins out. In parentheses, it says the fluid mosaic model. And the reason for this, people, students, they tend to think of, when they hear the term cell membrane, they tend to think that it's a very static structure, that it doesn't move. But that's not the case. Those phospholipids are able to easily change place with neighboring phospholipids. They're able to move and switch. It's a very fluid structure. So that's why it's known as the fluid mosaic model, to kind of have students keep in mind in the back of their head, that it's not a static structure staying still. It is constantly changing. It is selectively permeable, and we'll talk more about that on the next slide. Again, I like pictures, so we'll talk about what it's permeable to and what cannot pass through the cell membrane. Now, how it's different than a eukaryotic cell membrane is its role in the making of energy, or the making of ATP. So when you talk about something like the electron transport chain, I know you touched on this back in anatomy and physiology. In eukaryotic cells, the electron transport chain is located in the mitochondria. But bacteria cells don't have mitochondria. But they still have to make ATP, they still have electron transport chains, but it turns out that the electron transport chain is embedded in the cell membrane. Another thing, I don't want you to think that there's only one electron transport chain per cell. That is not accurate. Some bacteria cells will have 50 to 100 copies of the electron transport chain embedded in its cell membrane. You can also damage the cell membrane, just like we were talking about damage to the cell wall. You can damage the cell membrane by alcohol. This is the main way that hand sanitizer works. The active ingredient in hand sanitizer is alcohol. Alcohol works by dissolving the cell membrane. So you dissolve the cell membrane, thereby weakening the entire bacteria cell. Eventually that bacteria cell will lyse, or it will bust open, die, therefore it will die. So that is how hand sanitizer works. It works by dissolving the plasma membrane, or that cell membrane. We'll later on talk about quaternary ammonium compounds. These are industrial strength detergents that are very effective at damaging the cell membrane. Polymyxin antibiotics, again, an example of this is something like Neosporin. These work again by causing disruption of the cell membrane, causing the contents of the cell to leak out, and therefore the entire bacteria cell becomes very unstable, unsteady, it lyses and busts open, and then it dies. Okay. So let's put everything that I've kind of talked about here, let's put it in picture form. Let's focus on the bottom left picture first. So that is the phospholipid bilayer. Again, it's called bilayer because it's two separate layers of phospholipids. And you can see how they are oriented so that the heads face the watery environment of the outside of the cell or the inside of the cell, and the tails orient themselves so that the tails of both layers orient themselves towards each other. You can also see the integral proteins, and you can see the peripheral proteins. Now, if you were to take a small chunk of the cell membrane out, that is what you can see on the bottom right picture. So again, the bottom right. The head regions, this is where the phosphorus is held, or the phosphate. The head region is hydrophilic. meaning water loving. Okay, so the head regions always face near either the cytoplasm or the inside of the cell or outside of the cell in the watery environment. Whereas the tails are fatty acids. They are hydrophobic. They do not like water. So hydrophobic means water fearing. So they orient themselves naturally in this way so that the hydrophobic tails face each other, creating a hydrophobic region of the cell membrane. If you have small molecules that are hydrophobic and they need to pass from one side of the cell membrane or to the other side of the cell membrane, they can go straight through the cell membrane from one side to the other. So again, hydrophobic molecules can easily pass from one side of the cell membrane to the other side of the cell membrane. However, hydrophilic molecules, water-loving molecules, cannot pass directly through the cell membrane because the interior is hydrophobic. Okay? And water is like trying to mix water and oil. It just doesn't happen. So if any kind of hydrophilic molecule wants to pass from one side of the cell membrane to the other side of the cell membrane, it can only do that by traveling through pores, channel pores. And that is one of the functions of some of those integral proteins that you see. The integral proteins, they have several functions. Some function as enzymes, some function to help stabilize the bacteria cell, but some of them act as porins or pores. They're basically channels that allow hydrophilic molecules to pass from one side of the cell membrane to the other, since it cannot pass directly through the cell membrane. So in addition to the integral proteins, you also see the peripheral proteins. The peripheral proteins, these are proteins. Periphery means edge. Okay, so these are proteins that are found on the edge of the cell membrane. They do have, if you look at the picture, they do have like these curlicue structures that hold them tight in place, embedded in the cell membrane. But the peripheral protein, the protein itself, is not embedded in the cell membrane. Again, some of these function as enzymes. They function as support systems or part of the cytoskeleton to help give bacteria shape and strength. So the proteins have a variety of potential different jobs that they could have. The upper right-hand corner, this was a picture of a real-time picture taken with a transmission electron microscope where you can see the inside of a bacteria cell, and you can see that it is a gram-negative bacteria because I see an inner plasma membrane or inner cell membrane. I see a very thin layer of peptide glycan, and I also see an outer membrane. So by the presence of that outer membrane, that tells me that this has to be a gram-negative cell. Okay, so what else do we find in bacteria? So we are done talking about both the cell wall and the cell membrane. So what's left? To be honest, not much. There's really not that much inside of a bacteria cell. The majority of the inside or the interior of a bacteria cell is empty space. Okay. So on the inside of bacteria cells, you do find, remember they don't have a nucleus, but you do find what's known as a nucleoid region. This is the area where you can typically find that one chromosome. It is not membrane bound, which is what separates a nucleus from a nucleoid. Okay. The nucleoid region, again, that one circular chromosome, this is the majority of the genetic material. It contains up to about 3,500 genes, and is these genes that give the cell direction as to what it's supposed to do next. All right, plasmids. Plasmids are really cool, very important, but I'm not sure if you've ever really heard of a plasmid before. So a plasmid is DNA, okay? But it's a very, very small piece of DNA. It is circular. Okay. You can look at the picture here and you can see the plasmid. It is circular. It's very small. It is non-essential. What that means is that bacteria cell does not have to have the plasmid in order to survive. Let me also say, some bacteria cells have plasmids. Some bacteria cells don't have plasmids. We have made major advances in genetic engineering, where a lot of our genetic engineering today occurs on plasmids because they are easy to manipulate. You can take a bacteria cell that does not have a plasmid, and you can insert a plasmid into it. Okay. And so if you want that particular bacteria cell to do something, you can take the gene that codes for the production and insert that gene in a plasmid, take the plasmid and insert it into the bacteria cell, and then that bacteria cell will do whatever that gene is telling it to do. So when we were talking about insulin production in chapter 1, and I said most insulin today is produced through genetically engineered E. coli cells. A lot of that is done on plasmids. Okay. So plasmids are really cool. They're very, very important, but not all bacteria have plasmids. So I said they're very small. They only contain roughly 5 to 100 genes. They're non-essential, so you can take a bacteria cell, I think I mentioned this, but you can take a bacteria cell that has a plasmid, you can remove its plasmid, and the bacteria cell will still survive. Okay. And it replicates completely independent from the main chromosome. Okay. So it does its own thing. All right, so that's as far as the genetic material that you'll find inside of a bacteria cell. It will be found on the nucleoid region and then maybe on a plasmid if that bacteria cell has a plasmid. So other than DNA, what do you find in a bacteria cell? Well, moving on to the next slide. Again, well, there's ribosomes. So I mentioned earlier that the only organelle that bacteria have are ribosomes. And ribosomes, I mentioned, is the site of protein synthesis. There is 70S versus 80S. Prokaryotic cells have 70S ribosomes, whereas eukaryotic cells have 80S ribosomes. The ribosomes are composed of RNA and different types of proteins. Okay. And you can see the structure of a ribosome. It consists of 2 subunits. You have a large subunit and a small subunit. The large subunit sits on top of the small subunit. And then RNA comes through the top of the ribosome, and it gives or it yields a code for those amino acids to be made. And that's what a protein is. It's chains of amino acids. So that's a little bit as to how ribosomes work as far as making proteins. And other than ribosomes, you really only have inclusion bodies. Another name for inclusion bodies are storage sacs. They're basically vesicles to hold leftover nutrients, monomers, maybe there's simple sugars in there, there's amino acids in there. It's storage. It's like leftover storage. Okay. You may find some iron, as in this picture here. You see a magnotactic bacteria. It's a bacteria cell that has a lot of iron in it, and you can see those iron stores in the bacteria cell. So within these inclusion bodies, again, either iron or some type of leftover nutrient, sugars, amino acids, carbohydrates, lipids, something of that nature. So that's really all you find inside of bacteria. So let's kind of recap. Bacteria are very, very small. They don't have a nucleus. They have 1 chromosome, or typically 1 circular chromosome. They don't undergo mitosis or meiosis. The only organelle that they have are ribosomes and inclusion bodies, which are those storage sacs. That's really it. Bacteria are very, very simple when compared with any type of more advanced, any type of eukaryotic cell. Okay. All right, so the last 2 slides in this chapter deal with endospores. So we talked about endospores in the last chapter because we talked about staining endospores. If you remember, we stain them with malachite green, and you have to add heat because endospores are covered with a spore coat, which is a very tough structure. So you add heat and malachite green to stain those endospores green. I said in the last chapter that they are very, very hard to kill. They're dormant. They're very tough. There's a strong relationship with endospores being related to bacteria that cause disease. So to date, we've only found 2 genera of bacteria that produce endospores. That is Clostridium and Bacillus. I gave some examples in the last chapter of different diseases that are caused by Clostridium and Bacillus, but again, you have things like Bacillus anthracis, which is anthrax. You have Clostridium difficile or C. diff, Clostridium perfringens, which causes gangrene, Clostridium tetani, which causes tetanus. And there's several more. That was just a very, very small list. But the bacteria that fall under the genre of Clostridium and Bacillus can be very, very deadly. It doesn't help that these 2 genera of bacteria have the ability to produce these endospores. When these endospores are released into the environment, they can stay for many, many years. And they will stay in a dormant endospore state. And then there will be a trigger that comes along that tells that endospore, okay, it needs to germinate. And then when that's the case, it will germinate and develop into a parent cell. And then the process starts over. The parent cell will form an endospore, and the endospore will live inside the parent cell until the parent cell dies and lyses, and then the endospore is released back into the environment again. So it's basically a cyclic process, which I'll show you on the next slide. But you see that these endospores, they are, like I said, they're very, very hard to kill. They are resistant to UV light. They are resistant to gamma radiation. They're resistant to desiccation, which means drying out. They are resistant to lysozyme. They are resistant to temperature changes, extreme temperatures. They are resistant to starvation. They are resistant to chemical disinfectants. Like I said, very, very hard to kill one of these endospores. They're dormant, but they have very tough spore coats around them that help protect them. So moving on to the last slide in this chapter, this is the sporulation process. So if you look at step 1, the first thing that happens, so you start off with a parent cell, and you see it has a copy of the chromosome in purple. Well, it makes a copy of that chromosome and pushes that chromosome to the end, and then it starts forming a plasma membrane, and then a second plasma membrane in step 3, and then by 4, you form the layer of peptidoglycan. In step 5, the spore coat forms. And then in step 5, the parent cell or that host cell dies and it lyses open, releasing the endospore into the environment. And that endospore in step 6 can live on surfaces for many, many years. Some are even saying 30 to 40 years. Like I said in the last chapter, archaeologists claim they found endospores from hundreds, if not thousands of years ago, living in honey. And once those endospores were placed back in an appropriate environment, they germinated and they started to grow. So that endospore will get triggered. There's some type of trigger or signal that will tell that endospore to germinate, and when that happens, it goes all the way back to step 1. It will form a parent cell, and then the parent cell will start to produce a new endospore. So like I was saying, it's a cyclic process. Okay. All right, and that is the end of chapter 3.